Thanks again, Mike, for the introduction, and actually for introducing many useful, let's say, concepts that I will actually go a little bit more in detail. So you have seen, I think you've been convinced that using imaging allows us to understand deeply the underlying biochemistry of many diseases. And this actually helps us to find the most specific treatments. In basically, seeing is believing, and we know that from many years. But is your study, can your study benefit from radiopharmaceuticals? That actually is another question, and it may not be all the cases. So I see that there are three potential strategies, there may be others, but three main strategies to actually include in your development study radiopharmaceuticals. In the first case, you actually have a particular, let's say, target, and you have developed a molecule to be uptaken, but that to be accumulated in that particular target region. And this molecule might be a new molecule, so you're actually making a direct assessment of the population or a target of the special target in a particular subject. And it actually may include the design of a new radiopharmaceutical. But the other possibility actually was very well explained by Mike, was actually to make an indirect assessment. So you're developing a treatment, but you're actually checking the success of this treatment by using a standard pharmaceutical. And in this particular case, I personally find standard as a pharmaceutical that has been used by more than 5 different groups globally in the world. So in this particular example, for example, you see that actually there's been a tumor model, and then 2 different anti-tumor treatments has been used, and FLT has been used to monitor the success or not of this anti-cancer treatment. And you can see with your eyes which one worked. I just want to point attention here that the researchers decided to use FLT and not other tumor, let's say, imaging agent. Think about, for example, gallium PSMA, simply because, I mean, using this strategy means that you have to select properly also which standard pharmaceutical you want to use, in particular, a standard pharmaceutical that hits the target you're actually touching with a treatment. The third strategy that actually is used for tumors only is the use of pharmaceutical to deliver a radioactive payload on tumor cells, and basically to kill them. And actually is linked to the fact that we identify a vector that interacts specifically with tumor cells, and these vectors bring with it a radioisotope that have some killing characteristics. This actually, it's obviously it's important, but it's limited to tumors. So, sorry a second. I will not speak in this about the second, let's say, strategy, because I mean it deals with standard pharmaceuticals. There's a little bit more easy to get around. In all the cases, we need to make ourselves a few questions. So first of all, do we have a clear idea of which is the biochemical pathway that we want to study? And do we have an idea of which is the best molecule or class of molecules that is more useful to study this target? And do we have an idea of which isotope we want to choose to make the imaging of the treatment? And having said that, do we know how to radiolabel these structures? And further, we have this pharmaceutical. Has it been characterized properly at an in vitro or animal level so that you gain enough information to potentially go to the clinics? And talking about the pets, it's effectively a clinical, let's say, tool. So we have to understand a few of these things. And I will speak about some of these aspects in a separated way, but effectively all these aspects are intertwined. They don't exist in isolation. We cannot understand how to radiolabel a molecule if we don't know which molecule to choose, or we don't know which isotope to choose. So let's start speaking about, for example, the choice of radioisotopes. And mirroring what Mike was saying, this on the half-life point of view, this might be an easy choice. because if you actually, your molecule of interest is a drug-like small structure, it will actually reach a target in a matter of minutes or hours, and therefore we can label this with the short-lived isotopes. If instead we require to use large biomolecules like proteins, antibodies that require hours or days to actually reach a target, we need to label this with long-lived isotopes. So this rule of thumb, I would call it, does not account for some other aspects, like for example, if we can use a pre-targeting approach, but it's a good rule of thumb, and therefore we can actually go to the various options we have for radioisotopes. So in this table, you see most of the most used isotope used in imaging. I actually have highlighted the most used by far, technetium-99M, but it's not useful in PETs. I've highlighted in purple and in bold the isotopes that instead can be used in total body PETs. And in this particular field, we see the F18 still plays a very, very key role in PET imaging, followed potentially by gallium-68. There is a big speaking now in these days about targeted radionuclide therapies that requires a different kind of isotopes. And the difference between these isotopes is mostly due to which particle they emit, and therefore which kind of damage they can inflict to the tumors. So it's a very interesting field, and it's indeed, let's say, growing in importance, in impact, in applications. But none of these traces you see in this table is actually purple and in bold. None of these traces can be used in PET imaging. So why a person could be interested in so-called therapeutic radioisotopes? Well, because there's a concept of Theranostics radioisotopic pairs. So we can use the same molecular structure and then label it with one imaging isotope, or use the same structure and label it with an isotope that induce a cellular damage, a therapeutic isotope, as I call it. And we can do it basically using an iso-elemental approach in which the imaging isotope is the same chemical element of the therapeutic isotope, and you see there are some that we can use for PETs, or an hetero-elemental approach in which the imaging isotope is chemically different from therapeutic isotope. However, the chemical reactivity in this particular hetero-elemental strategy, it will be slightly similar, so we can use the same structure that we create the same different, let's say, isotope in as much as possible a similar way. So still, total body PET can actually have an impact to understand these theranostics, let's say, options. Now we have this nice pharmaceutical, and we want to understand which is the best, because we may have several, let's say, choices. And I will actually suggest you to go through this guidance for preclinical studies with pharmaceuticals published by IAA this year, because it gives you a very nice idea of the whole process, and also gives a nice information of what could be the desired features for the best pharmaceutical. In particular, a good pharmaceutical must have an uptake in the region of interest that actually it's useful in the times of the decay of the isotope. It has also have a minimal uptake in non-target regions, or at least it needs to be quickly washed out for those non-target regions. It needs to have an adequate decay stability, and the radiometabolites produced should be non-binding, so it should not stick in our target. Excluding the radiation damage that you want to induce, sometimes it must have a minimal toxicity or pharmacological effects, and importantly, it must have a viable radiolabeling and logistics. So when you speak of radiolabeling, I see mostly three routes to radiolabel a new structure. In the first case, we actually can have a structure in which we identify there is one particular atom of the structure that can be swapped for a radioactive atom. It can be the same atom, a fluorine-19 for a fluorine-18, or a different atom in a hydroxy group, OH group, or an F18. As long as we keep the pharmacological properties, we can do this operation. The other possibility, in the case in which this is not possible for many cases, is actually to modify the lead structure with a functionality they can actually react specifically with the smaller molecule having the partner functionality, typically using some click chemistry approaches. And this molecule here is typically called the prosthetic group because it brings the activity in the structure. But the possibility is still do on the lead structure modification, add a chelation group that actually can chelate some radio metals. So here are some examples. So this example of the racropide, you see in which the racropide bears effectively a methoxy group, and we have substituting one carbon with an adaptive carbon. And these are some examples of route 2 and 3. This was already shown by Mike, in which we have a desferoxamine chelating agent added to an antibody to chelate, that is able to chelate zirconium cations. These are examples in which we have added to a protein a little tag, a little appendix having a reactive tensioned cyclooctane that can quickly react and specifically react with a molecule containing an F18, but containing also an azyl group that actually reacts very easily with the triple bond. So when we try to implement these routes in our laboratory in Sweden, let's say, for example, we have once again to make some questions about the structure. Has the molecule that we are interested in been labeled before, or does it has a labelable position, or does it need to be modified to be labelable? And are there some requirement of, for example, molar activity? We speak a little bit about that later. About the regions, do we have the labeling isotopes of solution available on site or close enough? Are the chemicals, in particular the precursors for the labeling, available and well characterized and getting from a reputable source? Because sometimes we may have chemicals that may not be the purity required for the pharmaceutical process. And then the radiolabeling method, is the method robust enough, can be translated from one place to the other? Purification formulation also is known. Is automation be used, and at which level of reactivity? So in brief, there are a lot of information around the world. There's a global knowledge about how we can obtain these pharmaceuticals, but we have to put them down in our local capacity understanding if we can actually implement this knowledge locally. Because effectively, using molecular imaging and nucleomazine and total body PET is one tool for that. It allows us to understand our molecules and our treatments and the efficacy of them, and potentially allow us to understand quickly which are the successful routes and which are the not successful routes. So now we have this pharmaceutical in hand, and before thinking about total body PET, it would be extremely important to have a nice characterization, a preclinical characterization. So, I mean, I list here several of the things we can use for characterizing our pharmaceutical. It would be nice to have all of them, but in most of the cases, they are not all available. But it would be good to have most of them available. So, for example, we need to characterize this pharmaceutical in vitro, and we have to characterize the binding and internalization features, if it's blockable or if we can do some competition, if he has to cross some barriers, if we have some models to understand if the molecule crossed the barriers or not. We can do auto-radiography studies on tissue slices, tissue that represents the organ we are going to target. We can spend some time in understanding which might be the radiometabolites coming in and how to identify them, analyze them. And we understand if the radiopharmaceutical we have in hands is stable and is storable enough for our purposes. Then we actually go to the next level, an ex vivo characterization, in which we, for example, can inject the pharmaceutical in animal, and without undergoing imaging, we can just excise the organs and count them, so basically doing a biodistribution, or slice these organs and make autoradiography of the tissue slices. And we can also use the information of the previous, let's say, step to quantify how much of these radiometabolites are in the several organ districts. And we obviously need to do some kind of in vivo characterization using some animal models. we need to understand a little bit better the imaging protocol. And for example, injected mass and moral activity considerations are important. Also, specific assessment if in the in vivo model the drug remain compatible with the isotopology or with other gold standard ligands. And we want to understand a bit more the kinetic modeling, so do a little bit deeper imaging data analysis. At this point, we can actually go towards the clinical use and potentially the use of total body PET. So we have to think about a submission to human ethics, in which we will have to speak about many things, but one of them is dosimetry. And likely we can actually get some assessment of this from animal data and some algorithmic models. We need to understand the toxicology, and what helps in this particular field is that we can assume for pharmaceuticals the macrodosing scenario in which we inject less than 100 micrograms in the subject, and then few other things, typical imaging design, access scheme, and so on. An interesting thing to figure out very quickly, and I think that John will actually speak about that in the next talk, is how to organize the pharmaceutical supply. And one key point is to understand how much activity we have to inject in the person, what is the concentration of the dose, and if there are some molar activity requirements. And I get to this point because, I will not explain exactly what molar activity is. Molar activity is a characteristic of the pharmaceutical that tell us how good we are using the tracer principle. So once again, as a rule of thumb, a high molar activity will give us the possibility to get a good imaging without getting any pharmacological, let's say, effect. So typically, the highest molar activity is suggestible, it's preferable, but it's not always the case, because it may not be possible to get a very extremely high molar activity, or because in some cases it's actually not required or not needed. And I think that actually we have to think about, we have to create processes to make these pharmaceuticals, we have to validate our systems and quality control, and we have to understand if we are, in which way we are in the TGA exemption. So how total body PET helps all this process in understanding how to deal with your study of your pharmaceuticals in many ways. Well, the longer field of view allow us obviously to understand, interpret better off-target uptake and better tracer kinetic analysis. But the fact that we can inject less dose in a patient effectively lowers the pressure on the supply of the pharmaceutical. So we can actually use, accept lower efficiencies in the process. For example, we will have definitely lower dosimetry, that's a given, but also lower toxicity because we inject also less amounts of drug. And the widened scanning times may allow us to increase the portfolio of accessible tracers, because we could use short-lived isotopes in longer imaging times, or actually even have several days follow-up if you actually use longer-lived isotope. So there are many ways in which total body PET helps us in understanding which radioisotope we can use and in which way we have to organize our activity. And with this, I conclude, and I introduce, let's say, John, that we speak a little bit more in detail about how we can supply these tracers.